Sir, you can start, sir.
Okay, good afternoon, everyone. Welcome to the first live session for the course Next Generation Sequencing Technologies, Data Analysis and Applications. So we have covered first few weeks and we'd like to take any question and query from your side. If you have any question, please. Good afternoon, everyone. Welcome to the first live session for the course. Please feel free to type in the chat box, and then also you can write your question in the Google form. So that has been shared with you already. So please look at the Google form and please put your question there, or feel free to type in the chat box where I can see the question and then I'll be able to answer. So I think I can start with few questions that has already been asked in the forum. So one of the questions that I see that has been posted is how does BLAST work, right? What is the algorithm that BLAST follows? So just to clarify, we have also discussed in the uh, uh, course is that BLAST works by seed and extend method. So there are two steps, as you can probably understand now. So the first is seed, so which means you create a keyword and search for that keyword in the reference sequence or database. And then the second part is the extend, which means you'll found some matches with probably within first few seeds, and then you look at the neighborhood of those matches and see whether you can extend that match. Okay, so that's kind of the idea. So these seeds are then first mapped to the reference, and then there is extension for this after that seed. So I hope that answers the question without going into all the technical details which have been discussed in the presentation. The second question that has been asked is like, so we have in the presentation, in the course, we have discussed, right? There are different ways, different options that you have to give, that you can give when you are mapping reads against a reference genome or reference sequence. And we showed, right, if you use local alignment, you can then improve the alignment rate, right? So these are few things that you have to keep in mind. But then even after trying all those options, if you still have low alignment rate, what should you do? So the answer is, in that case, probably you are not working with the right reference genome, or maybe the organism or strain that you are using, the data from the organism or the strain that you have obtained, it may not be very similar to the reference genome or reference organism, right? So in that case, you might want to look into other reference strains or maybe related strains that might give you better alignment, right? Okay, so that is the only option that you have after you have tried out all the different local alignment, etc. When you are aligning your reads. So I hope that answer your questions regarding improving alignment, right? So the other question is, I think there was a question a few days back, is how do you actually set up parameters for trimming, right? So we have talked about adapter trimming using BP map package. There is a BPDuck tool that we use. So there are some parameters that you have to give for the trimming process, right? So the question is, how do you set your own parameters, right, for your own data? So to answer that question, there are some parameter values, typical parameter values that are given in the manual, right? So you can look up and see that in the manual, there are some typical parameter values that would be mentioned, but they might not work. This is not necessary that those parameters will work the best for your data. So what you have to do is use a trial and error, right? So you have to change the parameter values. So you try with the default parameter values and see whether those work for your data. Now, the question you might have is, how do you check whether that worked for your data? and I think we have talked about this again in the course, is that after doing the trimming, we do another QC using fast QC. Okay, so this will tell us whether the trimming actually worked, right, and whether trimming actually removed the adapter sequences in the example that we talked in the course. So this way, you can try your different values for these parameters and maybe try to understand which parameter kind of gives you the best results. So this is something that you have to tune if the default parameters do not work in your case. Okay, so here are some questions that I have answered. If there are other questions in your mind and or query, something is not clear, then please uh, send your question here. Okay, and also uh, type a question in the chat box, or you can also fill up the Google Sheet and type a question there, which we can discuss. So I see someone probably is going to type a question. So maybe I'll keep on discussing some of the questions that have been asked in the forum. Uh, so, so there's another question regarding mapping, right? In regarding specifically why do you do paired end sequencing, right? Why do you sequence the same fragment, right? So, and if we get the entire sequence, is it worth it, right? So the answer is, of course, if you get the entire sequence, if your fragment is small, in paired end sequencing, you will get both fragments. And then you're not getting extra information, right? You might think. But sometimes even then it could be useful because you might actually figure out if there is an error, sequencing error in the data, right? That's one of the advantages. The other is, of course, your fragments are often bigger, right? And you will not get the entire sequence from both ends, right? So in that case, you'll get only part of the ends. And even then, and it's very useful, right? Which I believe we'll have discussed, or maybe we'll discuss in the future classes, is that these paired data actually help in solving structural variants. Okay, so that's one of the advantages. The other is it also helps in resolving repetitive sequences. So if you are mapping repetitive data, right, repetitive sequence, that in that case, paired sequencing data is very, very useful. So I believe this has been discussed in the class. So there are other things I think some of you have raised is one is maybe someone tried the hands-on, like the C commands, etc. So some of these things you have to check carefully in Linux when you are writing any command or accessing any file. Please make sure that you are typing the right file name. Okay, so the correct file name is very important, and the file name is sensitive to capital and small letters. Okay, so this is something you need to be careful about. Okay, so this so far these are the main questions I have seen. Regarding reference books, some of you asked about reference books. This is mentioned in the NPTEL course page, and you can check some of them that are available there. Okay, so the other things, let's see. I think someone is making a question. Okay, so I think one question that has come from in the Google Sheet is that Illumina read length is 150 to 250 base pairs. So how the fragment size will be in this range for reading and mapping? Okay, so the question from Ismail is that if we get this 150 to 250 base pair reads, uh, how do we ensure that the fragments that we get from the genome, right? When you fragment the genome, we get fragments of this range. Is that a question? If not, please also type in the chat box, then I will answer that. So if I understand the question correctly, right, the fragment size, when we prepare genomic fragments, right, we take the genome, we isolate genome and fragment it through sonication or other methods, the fragment sizes are usually larger than 150 to 250. Okay, it's not necessary that all fragments will be within this range, some of these fragments will be much, much larger, like 500 base pairs, 1000 base pairs, right? Some of them will be 1.5, 1500, 2000, right? So we use a method which will give us around at least at most 750 to 1000 base pairs, because that's the limit for Illumina proper library preparation for bridge amplification. Otherwise, you tend to have improper bridge amplification. So the fragments can be of size, let's say 150, 500, 1000. Illumina will sequence all of them. Okay, so the only thing is that if your fragment size is, let's say, 1000 base pairs, you'll get only 250 from each end, right? So if you are using single end sequencing, you will get only 250 base pair out of that 1000. Okay, you will not be able to read the full fragment, only part of the fragment. If you use paired end sequencing, then it will be 2 into 250 base pairs. So you'll be reading 250 base pairs from both ends. Okay, so you'll read 500 base pairs, but even then you will still have 500 base pairs in the middle that will not be read. Okay, so I hope that makes the whole concept clear, is that the fragments that we prepare from genomes, they do not need to be within that range or in the range of read length. They can be smaller or bigger. Smaller will be covered very easily. For bigger ones, we'll get only part of the sequence. Now, there is another step when you prepare the library, which I believe we mentioned, is we also have a size selection, right? So we try to have only up to, let's say, 1 KB or so, or maybe choose between 500 to 1 KB. That's the ideal length for Illumina sequencing, ideal fragment length for Illumina sequencing. Okay, so I hope that answers your question. If not, please feel free to type, and then I'll be answering your question. Okay, so now there are some questions in the live chat, so I'll ask question. I'll answer those questions. So first question is from Ajit. So Ajit, so you have tried mapping for your own data, but you're not very sure about the reference genome, right? So you're saying you have tried to select the best one based on your morphological features in the page. So as you do the mapping, then you will see whether the alignment rate is good or not. Uh, so if your alignment rate is good, right, then you probably have chosen the right one. If not, you can do a search, right? What you can do, you can take a small part, right, of your genome, and or you can try, because it's a page genome. So for bacteria, what you do is, like, we can take the 16S sequence, and we can search against multiple organisms, right, and see which one gives the best match, and then we do the reference genome mapping, right? So in your case, because you are working with page genome, maybe you want to try also few related pages, right? Maybe that will help you, and see which one gives you the best alignment rate, right? So that will, in this situation, if you're not sure about the reference sequence, which would be the closest species, you might want to try a few, right? And a few related ones based on, again, your morphology and some other phenotypes, let's say. So that is something that, and then once you have done this mapping for a small fraction of each, perhaps not for the whole uh, genome data, then you can see which one is the closest, and then maybe choose that one as the reference. Okay, so it's a good idea to test out different reference genomes related from related species if you are not sure about the reference. Okay, second question. I think it's not a question. I think you're asking is whether okay, so Ajit, I think the second part Ajit has written is, like, you didn't get encouraging mapping results, only few get sharp match. So as I said, like, try few related species, right? Related bacteria pages, right? And see which one gives the best reference. Okay. Okay, so the next question, I think, is about how to learn programming. Of course, then you have to give some time, right? You start from the basics, and you have to spend time and do hands-on, right? For learning programming, no theoretical knowledge. Theoretical knowledge is not probably good enough, right? Even if you give a lecture, you have to start writing codes to doing very small tasks, and only then you will get can do this. So then the next question, I think, Lakshmi is asking, like, can you use time tool for the 16S RNA data from nanocode? So I have not used time for it. So to be honest, I have not used for nanopore data. So I'll not be able to tell which whether it should be and it should be able to handle that. So I have not tried. So I cannot really say whether that tool will be helpful here. Okay. So the other questions. I think there is another question. I think. Uh, okay. So the next question, I think again there is another question in the Google Sheet is. The reference and query mapping, I think, is by LF mapping, right? LF, so last pass mapping, right? Board I2. So is there any sequence size limit to read the sequence by this method? So if you're talking about the read sequence or the reference sequence, the answer is no. There is no sequence size limit. Nowhere there is any such limit imposed anywhere. Okay. If you're talking about reference sequence or about read length, there is no limit. You can use even longer reads. Uh, so that's not an issue. The only drawback, as probably have seen in the class when in the course when we discussed left mapping, last first mapping, is that if you have a lot of error, a lot of mismatch, there will be backtracking, right? So backtracking can increase the competition time and sometimes may not give you the best results, right? So that's one of the drawbacks, but otherwise there is no limit on the length of the read or on the reference sequence. So I think there is now another question in the chat. So Kabilan has one question is, so I think the question is, you are now analyzing or studying Illumina sequencing data for other sequencing platforms data. Do we have to follow the same procedure? Yeah, so the idea is exactly similar, right? The pipeline is very similar. You take the data and you analyze it, right? You do a quality control, but then again, it sometimes depends on the platform that you're using. Right? So for example, some platforms will not give you the quality score, right? It just set the quality score to 0. In that case, you cannot do anything. Uh, but then if the data is, if you have a quality score, we will go ahead with the similar process, right? We do a quality assessment. We can do the KMAT check, right? We can see GC content, etc. To validate that we are working with data from the organism that we wanted to, right? And then, of course, then you have to do the mapping and then all sorts of analysis that we talk about. The pipeline is very similar, only the first few steps, right? The data processing part, right? The data reading part, the format will be different. And you have to handle those different formats of data. Now, the good thing is this data can now be converted. Like most of the data formats can now be converted to FASTA format or FASTQ format. So if you can convert data from other platforms to FASTQ, then you can use very similar tools, right, that we used for Illumina or the tools that we discussed for Illumina sequence. Okay, so I hope that answers your question. So if there's any other question, I'm happy to discuss. Uh, so if you have any question, please type in the chat box or in the Google Sheet. Any question, query, any clarification required on any part. Or if you have any question on the regarding the assignments, so hopefully you are doing the assignment. So if you have any issue with that, then please please feel free to ask. So I believe you have also got the hands-on data, right? The hands-on that was demonstrated, and we have got the data and the code, and you can try on. They should be accessible, and you should be able to try it out yourself. Okay, because the hands-on actually matters rather than it's not just a theory. So hands-on will help you uh, get a feel for the data analysis techniques. Okay. And if you have any question regarding assignment or hands-on, then please feel to uh, ask. Okay, so there is another question in the Google Sheet is the question is how to choose the type of adapters for library preparation. Okay, so which type of adapter should you use for library preparation? Okay, so again, so the first thing is the adapters are platform specific, right? So your Illumina will use one type of adapter, PyPy will use another type of adapter, Nanopore use another type of adapter. I hope you get an idea of that when you see different technologies, right? Regarding within platform, right? Let's say in Illumina, we want to do DNA sequencing, RNA sequencing, small RNA sequencing, all those things. Do you always use the same adapter, or can we use the same adapter or any adapter for all of them, right? Or so does it need to be very specific? So the answer is yes. For Illumina, the adapters that you will use are very, very different. You probably have an idea, you got an idea about that, right? When you are doing the adapter, kind of looking at the quality control data, like for adapter training, etc. So these adapters are very, very specific, depending on the type of sample you have. If you're doing, for example, RNA sequencing versus small RNA sequencing, the adapters will be different. If you're doing genome sequencing for a very small amount of DNA, your adapter will be different, right? Because the principle of library preparation will be slightly different in each case, okay? And if the principle of library preparation is different, the adapters are likely to be different. So for example, if you are using a very small amount of DNA to do genome sequencing, so to prepare the library in Illumina platform, we'll use something called NextEra. Okay, so NextEra adapters. So again, without going into too much details, it's based on like that something like a transposon that will insert the adapter sequences in the genomic DNA and fragment it. So that's why the specificity counts, right? So the answer is yes, we need to be, these are specific when we are preparing the library because the methods of library preparation would be slightly different for DNA sample, RNA sample, and for example, small RNA samples. Okay. Okay, so regarding the textbook, again, I think there's some, uh, so some good textbook regarding related to this course. I think there's some textbooks already mentioned. Please have a look. In the course website, right, NPTEL course website, it's mentioned some of the textbooks. One thing I should also clarify that not all concepts that we are discussing are available in textbooks yet, right? Many of them are from papers. I also give references to papers that you have seen in the coursework. Because this is a new field and a very fast evolving field, right? As you see, this is not very old, right? Maybe 10, 15 years now, Illumina, etc. So that's why not all things that we discuss are available in textbooks. So some of the textbooks I have given, I have referred to, I have mentioned in the NPTEL course website. And then also I have mentioned some of the papers that where from which we see the algorithms. For example, this algorithm, what I do, or Barasuda transform-based algorithms. These are not discussed most of the time in textbooks. So this is from papers, right? So please, if you are interested in more details, you can have a look at the papers, and these are given as references at the end of the presentations. Okay. I hope that answers your question. So the next question from Lakshmi again is pipeline would be better for whole metagenome sequencing analysis. So I'm actually not going into the metagenome sequencing part. As you have seen in this course, we do not discuss metagenome sequencing, right? So I'll say like maybe that's for a different course, different time, and where we can discuss the metagenome part, right? So in this course, we do not discuss metagenome at all, metagenome sequencing. So I'll probably like stay away from metagenome part for now. Okay, so any further question from your side, please feel free to ask questions also regarding assignments and any query regarding presentations and reports, any confusion or any idea that is not clear to you. Okay, so maybe in the meantime, I can have a look at Forum and see if there are questions that has not been answered. Uh, yeah. Okay, so I think there was a question in regarding a bit earlier question regarding where to find the reference sequences. And I'm sure you have figured out now and you have seen where you find reference sequences, the NCBI website. So there is the site where different genome sequences are deposited, right? And you can find some annotation as well. Okay. All right. So I'm sure you have seen that. Otherwise, we can talk about this. Okay. Okay. So I think there is another question about assembly problem. So this is something actually uh, this will be discussed later, right? There is a specific part that actually is there regarding the assembly part, assembly algorithms, etc. Those will be discussed towards the end of this course. Okay. Okay, so in the Google Sheet, we have another question. So you're talking about so there is a question about whole exome sequencing, right? Is the concept same? And what are the major differences and challenges between whole genome and exome? So the idea is kind of same, right? Whole exome sequencing and whole genome sequencing. The only difference is that we are sequencing only the exons, right? And the intron regions and non-coding regions, right? Intergenic regions, these are not sequenced. Okay, so this is something we'll see in case of, for example, big genomes, right? For example, human genomes, cancer genomes. We have a lot of whole exome data and we do not sequence the whole genome. Okay, so this is something just to save on cost. Okay, so because whole exome, right, the coding region in human genome is like 2-3%, right? So we kind of save a lot on the cost of sequencing. Okay. Otherwise, if you have to sequence all bases, right, that will be much more expensive than whole exome sequencing. That's the difference. Otherwise, there is no other difference, nothing. Okay, so in whole exome, right, we are sequencing only a small part of the genome, so it's actually more economic than doing the whole genome sequencing. So this actually was done earlier because everyone thought or researchers thought that maybe is the gene mutations in the genes are that are very important, but recent understanding is that it's not only the genes, but also promoter sequences are important. And of course, we have now more whole genome data, for example, for cancer or human data, right? So that's the difference, nothing else. The pipeline, the whole process is same. The only thing is that cost and the mapping, the computational cost is also higher because you're dealing with more data and much bigger data sets. Okay, so I hope that answers your question. Uh, any other question? If you have, please type in the chat and uh, or post in the Google Sheet if you have any question, any query, any doubt. Or regarding the assignments, we have, I believe we have done 4 assignments already. Uh, and then I believe there is a problem solving session is also going on, right? So that might be there, but even then, if you still have some doubt regarding the problem sets, we can discuss. Okay, so in the meantime, if I can also see if there are other questions around in the forum. And so yeah, so I think one student mentioned that there is some sort of error when trying to do the uh, adapter trimming. Okay, so BB map, BB duct, the adapter trimming process, there's some error. And some of these errors could be because of Java. Okay, so maybe Java is not installed properly in the system. So maybe have a look, right? So either install the right Java version or update to the right version, right? Otherwise, you might get an error in the when you are trying to do the trim, etc. Okay, so that's the question. The next, I think, that is also a very interesting question. Is like, why don't we consider uh, duplicate reads in transcriptomics? Okay, so we consider duplicate reads and remove them when we're doing genome sequencing. We're working with single nucleotide polymorphisms or doing copy number variation analysis for genome. Why aren't we doing the same for transcriptome data? Why aren't we removing duplicates, right? So as you probably now understand is in RNA sequencing, right? Because we are sequencing only the coding region, right? Only the mRNA. So for genes that have very high expression levels, we'll often get a lot of duplicate reads. Okay. So that's expected. Okay. We expect to get a lot of replicate reads, especially for genes that have very high expression. Okay. So if we remove duplicate reads there, that will be completely uh, uh, changing the results there. That will introduce bias in the results. Okay, so that's why we do not do this in RNA sequencing. Okay, whereas in genome sequencing, it's not expected. Right? We do not expect a lot of duplicate reads. We just so because the fragmentation is done randomly at different location on the genome, so it's very unlikely that by chance two reads will start at the same position and end at the same position. Okay, so that's why for genome sequencing or for calling mutations, we remove duplicate reads, whereas for RNA sequencing, we do not do that. Okay, so there is another question, I think, regarding the landmarking in long read sequencing. Okay, so the question was like, what is actually landmarking, right? So this is we discussed when we do this in case of long read sequencing in Illumina platform, right? So we discussed that this is a synthetic long read, and there is landmarking, right? These landmarks are introduced in the fragments, and then the whole sequencing is done in short fragments. Then there is assembly using the landmarks, and then we get the long read data. But then the question was what are these landmarks, right? Actually, so the question is we do not know exactly what these landmarks are because the information is not released by the company, in this case Illumina. But there is speculation that this could be because they are introducing random mutations, or maybe they are introducing some sort of barcodes kind of thing in those fragments, and then they're using those mutations or barcodes to resolve. Okay. All right. So then let's go to the next question. So there is a question now in the chat. So this question is so the question is what are the important plots and graphs that we need for DMRS analysis and the correlation with DG's potent biomarkers in case of a particular cancer. I'm not sure that I think that there's some issue with the typing, etc. I'm not sure what you mean. If I understand correctly, it's like what are the important plots and graphs that we need to understand, right? So if you're when you are doing differential gene expression analysis. Is that the question? If not, please clarify. So if that question, so in case you want to identify potent biomarkers, right? So or correlation with disease to identify potent biomarkers in case of cancer, right? That. So of course, there are some flaws that are important for processing, like judging whether the processing of the data was correctly done, right? So we have talked about this, for example, the MA plot or the volcano plot. So those plots are used, right, in to actually judge whether, for example, MA plot will tell us whether normalization was proper or not. We can also look at outliers, right? And we can see what are the approximate number of genes that are showing differential expressions, etc. Now, the other thing, if you want to do, if you want to use this differential expression data as a potent biomarker, right, in case of cancer, right? So you see one gene, you want to identify certain biomarkers for, let's say, for certain cancer, right? And so what you have to do is you have to compare uh, normal versus cancer samples, and then you have to, of course, do the differential expression analysis and identify genes that show, for example, upregulation or downregulation compared to normal, right? And preferably a very high fold change, right, in expression. And then use those could be potential biomarkers for that cancer, right? So potential biomarkers, uh, the pipeline is same. We're comparing normal versus cancer, and then we kind of identify, or maybe we can use a ranking system to identify the ones that show significant difference, right? With adjusted p-value, right? We have talked about adjusted p-value, and then also the different the fold change also is very important. In addition to the MA plot or the volcano plot, we also sometimes use a heat map. It's kind of it gives us a good tool for visualization, right? We can visualize which genes are showing significant difference in expression, and also the magnitude of fold change, and that is something we can uh, do, right? So we can also prepare a heat map with normal sample versus cancer sample side by side, and we can then also look at some of these genes. We can identify some of these genes there. So this is again for visualization, and I think your question is like, what are the graphs or plots that might be useful in this kind of analysis? Okay, so I hope I answered that question. If not, please feel free to ask a follow-up question. That's absolutely fine. Okay, so I think if there's any other question here in the Google chat. So in the Google form, there is another question now, is there a difficulty associated with highly repetitive regions such as satellite DNA? And how can you resolve this by NGS, right? So that's kind of the question. So yes, of course, we have a lot of difficulty with highly repetitive regions, and which are really common in human genomes and other genomes, right? Highly repetitive genomes, these are very common. Repetitive regions are very common. So how do you resolve them, right? So what is the issue with repetitive regions, right? So the issue is this, like if you are working, for example, Illumina, etc., short-read platforms, is that if the repetitive region is longer than this read length, right? So if you get a read, you want to map it back to the reference genome, but this repetitive region maybe, since this is repetitive, it might be present in 5 copies in 5 different locations in the genome. So how do you know which region, which of these 5 regions this read comes from? So that's where the challenge is, okay? So if you have repetitive regions, your mapping is problematic, right? Especially for short read sequencing platforms like Illumina. For long reads, it's not a huge issue like Nanopore or PacBio, because you get much longer reads, which might cover the whole repetitive region, and you also get some neighborhood regions, right? Outside repetitive regions that will tell you, okay, this read comes from this specific location, okay? Because you have also covered some sequence outside the repetitive region, okay, which are unique. Now, For short read sequencing like Illumina, one strategy is to use the paired end sequencing, right? Because paired end you can generate fragments and you are sequencing reads from both ends, right? So there is a possibility that one end might fall outside the repetitive region, right? So if one end falls outside the repetitive region, you can pinpoint the exact location of that repetitive region. But if the repetitive region is really large, like few kbs, then of course by Illumina short read sequencing, you will not be able to resolve this. And your only solution is the long read sequencing platforms like Nanopore or SMRT. And in those cases, so when we're working with genomes of that kind of repetition, right, where we have a lot of repetition, and when to assemble genomes or map genomes, right, map mutations, etc., we often use long read sequencing along with short read sequencing, like Illumina, right? So this is called hybrid assembly. So why are you using both? Because long read sequencing, for most cases we have seen, they are not very accurate so far. So there is scope for improvement. And if you use only long read data, your SNP calls, the mutation calls may not be accurate. So what we do is we use the short read data like Illumina, which are very, very accurate, to correct the long read data for mutation calls and SNP calls. And then we can assemble, okay, and we can resolve these repetitive creatures. Okay, so with the advent of long read sequencing, we can now resolve this quite comfortably. And we can combine both short read, long read together, and then we can get a nice picture of the whole genome. So I hope I answered the question now. Okay, so the next question, I think from Kabilan, is can we practice the same procedures uh, uh, can you practice the same procedures for single cell omics data analysis? If not, what is the basic difference in analysis between normal omics data and single cell omics data? Okay, so that's a question for the future, I think. We have not discussed single cell omics data, or single cell sequencing data, RNA sequencing data especially, which is coming up now big place. So one of the I'll just end with saying that the analysis pipeline is not same. The reason for this is in single cell we have a lot of zero values. In single cell RNA sequencing data, we miss a lot of data, and we need to take that into account. So this is something that's very important in case of single cell data. And this is an important challenge. This is a very important challenge for single cell data. So this is, we need to discuss in a separate topic, right? It's not, I'll not discuss here because we have not talked about single cell data. So the next question from Sudha is like, is there any other way to present the correlation of the MRs with DGs other than the Pearson correlation method? Yes, so there would be other ways. So, I'm not sure what you want to represent. I think this is something that again is important. So if you are talking about biomarkers, right, you identification of biomarkers, so I think just beyond differential expression analysis and identifying those genes, right, those are potent biomarkers or potential biomarkers. You also want to look at something, you might want to also implement something like a machine learning method kind of thing, okay? If your goal is to identify biomarkers, right? I'll not again go into that. So for correlation, of course, you just look at Pearson correlation. That's probably Pearson or Spearman. Spearman is a bit robust to variation in the actual value, whereas Pearson is more sensitive. So again, the correlation is something that's again, I think the techniques will change if what you want to depict or what you want to show or what you want to identify. So that there are a lot of techniques that can be used, but then again, I think this will depend on your application. Okay, so I'm not sure like what your specific applications are. I'm not able to give you a very specific answer. But I hope that's clear. But if you have any further question, please feel free to ask. Okay, so if there's other questions, so let's, I'll look into the forum again and see if in the meantime, if you have any question, please type in the Google form or in the chat, right? So I can see and I'll be able to answer. So in the meantime, I can look into the forum and see if there is any other important question that I have not answered or just something is not clear. That I think I have discussed most of it. So I think also there's a question from seed and extend. So I just mentioned earlier that there is a blast uses seed and extend. So what is seed and extend? I have also discussed, right? So in the neighboring regions, we actually where the seeds match, we look into the neighboring sequences and we also try to find whether there are other seeds matching in the neighboring sequences. In that way, we can consolidate the mapping or alignment, and that will give us the final alignment and mapping. So I hope this is clear. So if there is any other question, I'm happy to answer anything anywhere. So please feel free to ask in the Google form or in the chat box. So I'll be happy to answer. Or if you have any question regarding the assignments or problem solving sessions, please feel free to ask as well. So again, I can look into the forum, right? Also, okay, so there was one question about what are the adapters made of, right? And what is the usage, right? Maybe if you have checked the forum, I have answered this there already. Is that so adapters contain index sequences primarily and also primer binding sites. So I have explained in the course is that primer binding sites, this is where the sequencing primers will bind, right? For Illumina platform where you need sequencing, where you need the sequencing by synthesis process. So we have polymerase, so that will synthesize the complementary strand and in the process will generate the sequence data. So you need a primer binding site. So the adapters contain a primer binding site. In addition, it contains index sequences. These are like something like barcodes, right? These are unique combination of a TGC, right, that has been designed to actually help mixing of samples, right? So if you want to sequence, let's say, 8 samples in Illumina, and you do not need a lot of reads for each sample, what you can do, you can mix those 8 samples and run in one flow cell and get the read, right? So now problem is if you mix them, how do you identify which read comes from which sample? This is where the index sequence helps. Okay, so index sequence is a unique sequence that is added to each sample. So all reads that are generated will have this index sequence. Okay, and once you have this index sequence on reads, you can then do the run, and based on this index sequence, you can identify, okay, this index sequence belongs to this sample, so it means this read belongs to this sample, right? So you can identify, you can do this mapping from index sequence, right? So the process of adding index sequence and mixing samples is called sample multiplexing, and getting the sample data out from the final read data for different samples. You identify different samples from the read data is called the demultiplexing. So this is something I believe we have discussed in the course. Okay, so I hope I have answered all your questions. If you have any further query, please feel free to ask, and I'll be happy to answer any query, anything, any question regarding the course, regarding your assignments, etc. Okay, so the next question in the chat box is from Sudha is for biomarker identification. So we have discussed the biomarker identification process. Is it compulsory to take genes that fall in the coding region only, or it's fine that we take not like introns or intergenic regions? Okay, so if you want to use a biomarker, right, based on expression, it will be a gene, right? Expression of a gene. If you want to use a biomarker based on mutation, right? It could be anywhere. It could be intergenic region, right? So, for example, for some of the cancers, there are biomarkers, right? We know some of the mutation patterns in certain genes, so we can use mutation patterns, right, as biomarkers, and in that case, it could fall anywhere. Okay, but if you're talking about gene expression based biomarkers, which is a common biomarker in many cases, in that case, it would have to be gene, right? You're measuring expression of the gene. So that's it. All right. So I hope I answered that your question regarding the biomarker identification. So any other question, any other query from your side? I'm happy to answer if any question regarding assignments, etc. So you can put a question in the Google form or also in the chat box, and then we'll be happy to discuss. Okay, so so in the meantime, I can also one more question I've seen in the forum I can discuss. So the question is like, how do you know whether the fragments are of a particular size, right? So is there an agarose jelly to persist right out after fragmentation, right? So when you do the genome fragmentation, we usually do not have control over the fragment sizes, right? So we get random fragmentation at different places. And after that, for library preparation, we do a size selection experiment, right? We have talked about this in Illumina platform. We have a size selection experiment, so we choose DNA fragments within a specific size range so that we get optimal sequencing results. Okay, so as I said, like in Illumina, when you're doing bridge amplification, there is a size range for which this method works the best. Okay? If you have bigger fragments or very small fragments, then this method does not work very well. So in that case, your data will not be good. So there is a size selection step in between before we actually proceed for library preparation and sequencing. Okay, so I believe the question. So there is another question is uh, so one is like one question that I think we might also maybe thinking about is since there are many improvements on WNT Oxford nanopore technologies and the error rate is now reduced, why do we still rely on Illumina? Can we employ both together? Like, can we not use both, right? WNT and Illumina, so Oxford nanopore and Illumina, right? So the answer is yes, of course, you can use nanopore for RNA sequencing, and this is applied when you have long RNA sequencing, right? Otherwise, Illumina results are good enough because RNA molecules are not really that long and they are not usually repetitive, right? So that's one of the things, that one of the constraints we have. So Illumina results are already pretty good, okay? And even with improvements, Illumina still has higher accuracy overall than the Nanopore platform, right? So if you're interested in also looking at, for example, some of the mutations happening in RNA, right? So if you have to study RNA molecule mutations or errors in the transcription process, etc., you will want to rely on a method that's more accurate. And in this case, it's the Illumina, right? So Illumina data is good enough and it gives more accurate read sequence. Okay. So I hope that answers this question. Right. So any other question? Maybe we can discuss if you have anything in your mind. Yes, so if you have any question, you can put in the chat box. Here also in the Google form. And let's see if we have any more question in the forum that has not been discussed. I believe I have discussed all questions that are there in the forum. I have answered all of them. So even then, if you have further questions, right, I'm happy to clarify. If you have any queries, some clarification required regarding the course, regarding the assignment, I'm happy to discuss. Okay. Any further question? If there is anything that you want to ask, and of course there will be more live sessions, right? Uh, three live sessions are scheduled. This is the first one, and after that there will be live session 2 sometime in September, and there will be live session 3 uh, during October, right? Of course, before the final exam. So if you have any doubt, any query, please feel free to ask there, right? In the forum, of course, I can type a question. Uh, uh, yeah, so that's from my side. Yeah, so now there is another question in the Google chat box is that so whether this course, the pipeline that we are learning, would it help in analyzing bacterial genome? Yes, of course it will. Any type of genome you can analyze with the tools that we have talked about. So the task may be a bit simpler in case of bacterial genome and may not be as computationally intensive uh, as for big genomes. So otherwise, the pipeline is very simple. Excuse me. Yeah, so hopefully that answers your question. Yeah, so if you still have further questions, I can answer. Uh, yeah, so there'll be more live sessions in future, live session 2 and 3. If you still have doubts, then we can discuss and queries there also. In the meantime, there is the forum, so please post your doubt in the forum and we'll answer. It may not be immediately, but we'll answer within a few days. Within a couple of days, that's what we try to answer all the questions in the forum. So post in the forum any doubt, anything. And of course, if you have something very detailed questions, very detailed query, that can also be taken up in the live session. So please feel free to post your questions in the Google form or in the chat box during the live session. So, and I'm happy to answer all questions. Okay, so I believe I have answered all the questions in the forum so far. Also, there is no other question in the Google form, and there's okay, there is um, okay, so I think Kavilan has a question that you have missed the problem solving session of week 5. So link of that session is not in the portal. So I'll request you to reach out to the NPTEL team, right? NPTEL office. They might be able to help with this. I have no idea about the links of how they're recorded, when they're put in the website. So please write to the NPTEL office regarding that, and they'll be able to help you. Okay, so if you have any further question, please feel free to ask. Otherwise, I think I have answered all the questions in the forums. But still, as questions come up, please feel free to post there. Would answer questions regarding whatever we are discussing in the course for, or in the assignments, or in the problem solving sessions. Any query, any doubt, please feel free to post. I'll be happy to answer. And we will also take up more questions and maybe some detailed discussion uh, regarding uh, questions you might have. The other question, Dr. Rudresh, you have is, can we use the same tools for bacterial genome analysis? Yes, we can use the same tools. The process does not change if you, for example, the pipeline for genome sequencing data analysis will not change depending on the organism that you are working with. Right, of course, the reference sequence will change, etc. Some of the things might change, the quality thing, etc. But the tools that we used in the course, they will not change. They will remain the same. Okay. So whether it's bacteria, yeast, fungus, human, mammalian system, or plants, if we're doing genome sequencing, the tools remain the same. Only thing is the computational requirement, right? So bacterial genome is smaller, so computational cost or computational requirement would be lesser than much more complex genomes. For RNA sequencing, again, the tools remain the same. But since the mammalian system, there are more genes, varying range of expression, right? So then, of course, you need to generate some data. But the data processing pipeline, they remain the same. We change in the reference sequence, etc. And for higher organisms versus bacteria, if you're doing if you're analyzing RNA sequencing data, since bacteria or maybe yeast, you have very so for yeast, so for these organisms we have no introns or very few introns, right? In that case, you can simply use MOTI2 for RNA sequencing, mapping of RNA sequencing data. But again, the pipeline, you do not need to change the pipeline. You can use the same pipeline. Things should be simpler for bacterial. Yeah, I hope that answers your question. Any further question, please feel free to type, and I'm happy to answer. Okay, so if there is no further questions or queries, then I believe we can close now. Uh, Please feel free to post your questions in the forum, and also we'll take up more questions in the next live session, which will be in September. That date has not been fixed yet, but that will be announced in the forum. So please make sure you follow the forum and follow these dates. So you can post your questions there, and you can follow the tape for the next live session where we can discuss further questions, or if you have some detailed questions regarding the covering or any problem with your hands-on, like what could be the issues, etc. I can tell you some possible solution. Yeah. Okay. So if there's no other question, I think we can close for today. Thank you.
Thank
Oh,
you, sir.
so there is, I think, one more question, right? So I think one last question I think we can take is that ONT platform is claiming 99.5% accuracy, and we have to work on bacterial genomics. Will it be sufficient? Yeah, yeah, of course. It will be 90. If it, so, ONT platform, I'd say like if they're claiming 99.5% accuracy, please check which chemistry they're talking about, what kind of accuracy they're talking about, like whether it's per read accuracy or it's the consensus accuracy. Make sure you understand those details. Okay, so usually, even now, when the per read accuracy is still pretty not in the level of Illumina. Okay, so that is one of the issues we have. Uh, Per read accuracy. Okay, so Illumina, where is it around 99.9%, is still not there. 20. So please make sure whether it's per read accuracy or consensus accuracy. Consensus accuracy will be above 98.5, etc. This might be important depending on your application, right? So in that case, yeah. So again, if you just want to look at the genome and assembly, if you have enough coverage, then yes, it will be sufficient. But if you want to, for example, identify variants from the genome, like you are sequencing a microbial population, you want to identify specific variants in that, which are present in different frequencies in the population, then your per read accuracy is more important, okay, not the consensus accuracy. If your idea is just to assemble the genome, then it's okay. Okay, so I hope I answered the question. Okay, thank you very much. I hope you find this course helpful and help with also the hands-on, right? So that I think is more important, and on top of the theoretical discussion. Okay, so I hope I answered all your questions, but if you still have some doubts, please feel free to post. Uh, Thank you, Dr. Rudresh. So please feel free to post your questions in the forum, and also we can take up also some of the questions in the next live session, which will be in September. Okay. Uh, thank you very much.